Welcome to G Fun Facts Online. Today, we are going to dismantle everything you think you know about the biological definition of life. If you have ever looked at a biology textbook, you are familiar with the standard cast of characters that make up the microscopic world. We have bacteria, the single-celled organisms that can be our best friends or our worst enemies. We have viruses, those genetic hackers that hijack cells to replicate. And perhaps you have heard of fungi or protozoa. For decades, science has operated under the assumption that while we might find new species within these categories, the categories themselves were settled. We built a tree of life, and we were confident in its branches. But in early 2024, that confidence was shattered. A team of researchers from Stanford University, led by Nobel laureate Andrew Fire and doctoral student Ivan Zelidov, discovered something that should exist, buried deep within the genetic data of the human microbiome. The very bacteria that live inside you right now. They found a biological entity that defies classification. It is not a virus, it is not a bacterium, it is not even a standard viroid. It is a completely new class of life, or life-like matter, that has been living inside us for millennia, entirely unnoticed. They call them obelisks. Over the next 20 minutes, we are going to explore how we missed an entire empire of biological agents hiding in our own mouths, what these obelisks actually are, and why their discovery might force us to rewrite the history of life on Earth. To understand the magnitude of this discovery, we first have to understand how it was made, because obelisks were not found by a biologist trekking through the Amazon rainforest or dredging the bottom of the ocean. They were found in the cloud. Modern biology has moved from the petri dish to the server farm. When scientists study the human microbiome, they take samples, swabs from the mouth, the skin, the gut, and they sequence everything. They turn biological matter into strings of letters, A, C, G, and U, representing the genetic code of RNA. This process generates petabytes of data, billions of strands of genetic information. Usually, researchers take this data and compare it to known libraries. They ask the computer, show me the sequences that match E. coli, or show me the sequences that match the influenza virus. They find what they are looking for, and they ignore the rest. The rest is considered noise or junk data. But the Stanford team did something different. They decided to look specifically at the noise. They asked, what is in the data that doesn't match anything we know? To answer this, they built a specialized computational tool called Vinome, which stands for Viroid Nominator. They weren't looking for specific genetic sequences, they were looking for a shape. They programmed the algorithm to sift through 5.4 million datasets of genetic sequences, looking for circular loops of RNA that fold in on themselves. When the algorithm finished its work, the results were not just a statistical anomaly, they were a revelation. Vinome didn't find a handful of unknown things. It found nearly 30,000 distinct types of a previously unknown biological agent. These agents were composed of RNA, much like many viruses, but they were incredibly small, only about 1,000 nucleotides long. To give you a scale, a typical bacterium has a genome of millions of nucleotides. But the most striking feature was their structure. When these RNA strands exist inside a cell, they twist and bond with themselves to form a tight, stiff, rod-like structure. It was this distinct, monolithic shape that earned them their name, obelisks. So what exactly is an obelisk? This is where the biology gets fascinating, because obelisks break the rules we have established for the borderlands of life. Until now, the spectrum of simple biological agents had two main poles, viruses and viroids. Let's define those quickly to see why obelisks are so strange. A virus is essentially a piece of genetic material, DNA or RNA, wrapped in a protein shell called a capsid. It is a package. It infects a cell, unpacks itself, and uses the cell's machinery to print copies of itself. It is complex enough to carry the blueprints for its own shell. On the other end of the spectrum, discovered in the 1970s, we have viroids. Viroids are mostly found in plants, like the potato spindle tuber viroid. They are tiny, naked loops of RNA. They have no protein shell. Crucially, viroids do not code for proteins. They are essentially parasitic information that disrupts the host cell just by existing and gumming up the works. They are glitches in the biological software. Obelisks sit in a previously impossible middle ground. Like viroids, they are naked circular RNA loops with no protein shell. They don't have a capsid. But, like viruses, they contain genes that code for proteins. This hybrid nature is unprecedented. They are small enough to be simple chemical replicators, yet sophisticated enough to manufacture their own tools. The proteins that obelisks code for are a complete mystery. The researchers named them oblins, specifically OBLIN1 and OBLIN2. When the scientists took the genetic instructions for these oblin proteins and compared them to every known protein database on Earth, They found absolutely zero matches. There is no homology or evolutionary similarity to any known biological agent. This is profound. It suggests that obelisks are not just a mutated version of a known virus. They are likely a distinct phylogenetic group that has been evolving parallel to us and parallel to viruses for perhaps millions of years. Initial structural analysis suggests that the Obelisks protein might have the ability to bind to metal ions, which could play a role in how the obelisk replicates or signals within a cell, but its true function remains one of the biggest unanswered questions in microbiology today. Perhaps the most unsettling aspect of the obelisk discovery is not what they are, but where they are. If these were rare entities found in a deep-sea hydrothermal vent, it would be an interesting footnote. But they are not. They are in us. The Stanford study analyzed human microbiome samples and found that obelisks are surprisingly prevalent. In samples taken from the human mouth, obelisks were found in approximately 50% of the datasets. That means if you are sitting in a room with one other person, statistically, one of you has obelisks replicating in your saliva right now. They were also found in about 7% of stool samples, indicating they can survive the journey through the acid of the stomach and the enzymes of the digestive tract. But obelisks are just RNA. They cannot float around in the air or swim on their own. They need a host cell to replicate. Through some incredible detective work, the researchers identified at least one definitive host, Streptococcus sanguinis. This is a very common bacterium found in dental plaque. This is a crucial distinction. Obelisks do not infect human cells directly. They infect the bacteria that live inside humans. They are parasites of parasites, or perhaps commensals of commensals. They are a hidden layer of the ecosystem, living inside the bacteria that live inside us. Interestingly, the presence of obelisks does not seem to kill the bacteria. The Streptococcus sanguinis bacteria that were hosting obelisks continue to grow and divide. This suggests a relationship that is not like a deadly viral infection that explodes the host cell. It seems to be more commensal, meaning neutral, or perhaps even symbiotic. The obelisks are quiet roommates, replicating silently without burning down the house. This silence is exactly why they remained undetected for so long. They don't cause obvious diseases, so we never went looking for them. The discovery of obelisks has set the world of evolutionary biology on fire because it reinvigorates one of the most significant debates in science, the origins of life, specifically the RNA world hypothesis. The RNA world hypothesis suggests that before DNA and complex proteins existed, life began as simple RNA molecules that could do two things: store genetic information and catalyze chemical reactions. For a long time, viroids, those simple plant pathogens, were thought to be the living fossils of this ancient era, remnants of a time when life was just naked RNA. Viruses were seen as later, more complex evolutions. Obelisks sit perfectly in the middle of this timeline. They might represent a transition state in the evolution of life, a step where simple RNA replicators began to develop the ability to code for proteins, eventually leading to the complex cellular life we see today. Are obelisks ancient survivors from 4 billion years ago, protected inside bacteria through the eons? Or are they a newer evolutionary experiment, a de-evolution of viruses that ditched their shells to travel light? The distinctiveness of the oblin protein suggests the former, an ancient, separate lineage that has survived in the shadows of the microbial world. Now, the million-dollar question for us as humans is, do obelisks affect our health? As of today, there is no evidence that obelisks cause any specific disease. However, in biology, absence of evidence is not evidence of absence. The history of medicine is full of harmless entities that turned out to be pivotal in health and disease. Even if obelisks don't kill cells, their presence involves producing RNA and proteins inside our gut and oral bacteria. This takes energy and resources. It could alter the behavior of those bacteria. For example, an obelisk might turn a harmless bacterium into a more aggressive one by changing its gene expression. Or conversely, it might prevent a pathogenic bacterium from causing harm. We know that our microbiome affects everything from our digestion to our mental health via the gut-brain axis. If 50% of people have obelisks in their mouths and 50% don't, there may be a subtle but widespread difference in oral health between carriers and non-carriers. Could obelisks be a factor in tooth decay, gum disease, or even resistance to antibiotics? We simply don't know yet. There is also a fascinating therapeutic potential here. If obelisks are efficient at entering specific bacteria and producing proteins, they could theoretically be hijacked by scientists. We could engineer designer obelisks to deliver therapeutic payloads into specific gut bacteria. Imagine using a modified obelisk to turn off a gene in a bacteria that causes inflammation, effectively rewriting the microbiome to cure diseases. This is a concept known as phage therapy. But obelisks offer a much smaller, potentially more agile vehicle for these changes. This discovery heralds a new era of computational microbiology. We are no longer limited by what we can grow in a petri dish. We are now exploring the dark matter of biology, the genetic sequences that don't match anything in our textbooks. The Stanford team's use of the Vinom tool is just the beginning. Researchers worldwide are now rescanning their old data, looking for other cryptic elements. If obelisks went unnoticed for so long because they didn't look like viruses, what other shapes are hiding in the data? Are there triangles, spheres? Are there RNA agents that live inside our own human cells, not just our bacteria, regulating our genetics in ways we don't yet understand? Biological obelisks challenge our rigid definitions. They are not quite alive, yet they evolve and replicate. They are not quite viruses, yet they are infectious. They represent a third state of biological matter, minimalist, efficient, and ancient. As we stand on the precipice of this new frontier, one thing is certain. The microscopic world is far more crowded, complex, and mysterious than we ever imagined. The obelisks have been watching us from the inside for millennia. Now, finally, we are looking back.